coordination control, there's three subtopics in this chapter. We have firstly the part which compares the nervous system to the endocrinal system, which we will do at the end of homeostasis, so it's kind of cohesive. And then with the main bulk of this chapter, which is how do action potential get generated and how they are transmitted and how do they work on muscles, like in general how the nervous system works. Okay, and then the last part is the part about the plants, which is like a little part about plant coordination, which we'll do at the end. But this is like the main topics, and questions usually come here. Does not mean that you should neglect the rest, because sometimes they do not get a lot of questions here, and then they end up asking here and here. So because of students sometimes leaving them, they lose marks on such easy parts. So let's start off with the nervous system part. Okay, system in a nutshell makes us think about how do we sense something, and then how does this sensation get transmitted in our body, and then how do we do an appropriate reaction to it, and how do our muscles respond. So this is everything in this part about it. The problem is we take each one of them as a separate thing and we don't think about it as a whole, so it kind of gets confusing and you don't know what are we just studying. But if we thought about it, okay, in just some steps, we can just understand everything that happens. So firstly, we have some sort of stimulus that happens. For example, let's say something pricked our finger. Okay, so we have to understand how do we feel this? How do receptors detect this pin stinging? Okay, and then how do those receptors send this impulse to our neurons? Okay, and how do those neurons respond? And then how does one neuron send this impulse to the second one? That's basically the synapse. Okay, and then how does this sensation go to a muscle, and how does this muscle do an appropriate response to this? So this is everything we're going to study in a nutshell, and this is helpful because it will make you kind of imagine everything. I'm going to revise on this part quickly so you kind of get the understanding of it, and then we're going to solve, and I'll re-explain again the parts when solving and then tell you how to think about it. Honestly, this chapter is an easy chapter. A lot of students have a problem with this, but if you genuinely understood this part really well and you solved and you kind of figured out the ideas that come, it's going to kind of seem repetitive to you, so don't worry about this. This first part in the spec is describe the structure and function of the sensory neurons, the motor neurons, and the intermediate neurons. We're going to skip this one first because there's a question coming on this one, but just so you know, so obviously this is a whole reflex arc. Okay, so this is the sensory neuron. Okay, and it has a specific shape where the cell body is in the middle, and it has a shorter axon and a dendron. Okay, and the sensory has the sensory receptors as its start. Okay, so here is where all the magic happens. Here is where the receptors detect your stimulus. Okay, then this sensory neuron send the impulse to the relay, which is in the center. This relay is usually in the spinal cord, also in your brain, but here the example we have is in the spinal cord, okay, which is called the CNS. Okay, then this relay sends the impulse to the motor, and the motor is connected to your effector, which is a muscle or a gland. Okay, so the question is, how do those receptors detect the stimulus? So this is the second question here, is outline the role of the sensory receptors in detecting the stimulus and the transmission. And the example we have is the chemoreceptor example. So let's look at this receptor here. This is a type of receptor. Let's call it a pain receptor. Okay, we have a receptor for each sensation. Okay, so the pain receptors detects any sort of pain. So let's say we got pricked by a pin. Okay, so when you got pricked by a pin, okay, here we have a kinetic energy. Okay, your body does not understand kinetic energy. So the role of this receptor is to translate, or in other words, transduce this energy from whatever type it is into something our body understands, which is electrical energy. Okay, so this is the function of the receptor. Number one, it's a transducer. So for example, the receptors in your eyes, they transduce the light into electricity. Your sound, the sound waves, into electricity. So any sort of energy is transferred into electrical energy. Okay, how does this happen? When this stimulus happens, okay, this causes some sodium channels in the cell membrane here to open. Once those channels open, sodium ions diffuse in. Okay, why does that happen? Because when we think about our nervous system, our axons and stuff, okay, so inside is more negative than outside, okay? Something else is that inside we have potassium, while outside we have sodium. Of course, potassium is still positively charged, but like relatively more potassium inside than outside, and sodium is more outside. So this is something we have to keep in mind all the time. This is at resting. So this is when you're just, nothing is happening to you. This is how your body is. Okay, so once there is some sort of change, those sodium ion channels open. That means because there is a gradient right now, it's called an electrochemical gradient, because you have both an electrical gradient where outside is positive and inside is negative, and there's also a chemical one because outside there is more sodium than inside. So the sodium has no option but to diffuse inside. Okay, so once it diffuses inside, this forms something called a receptor potential. Okay, there is a change right now. Inside is starting to become more positive. But like, to look at the curve right here, we have the curve, okay? And let's say that this is our normal resting, which is at negative 60 millivolt, okay? So normally our body is at this level, okay? Once there is some potassium entering, the negativity starts to decrease. So it's starting to get negative 59, negative 58, negative 57. Okay, it's starting to get more positive. Okay, till it reaches something called a threshold. Okay, this threshold is a specific number. Okay, according to each neuron. But here we are, like in our curriculum, it's negative 55. Okay, once we reached this negative 55, now everything happens. Now the action potential starts. Okay, so as long as we're below this negative 55, nothing will happen. The receptor potential will disappear and you will not sense this. Like, this happens when you get pricked or something like sticks to you and you do not feel it. Why? Because it wasn't strong enough to make you feel it. After we have the receptor potential, how do we generate an action potential? Okay, so if the receptor potential, as we said, is strong enough to exceed the threshold, all the potassium ion voltage gated channel in the axon will open. Okay, so if we took a section of here and then zoomed it in, okay, so let's say here this part of the membrane reached the threshold, which is the negative 55. Okay, so that means that enough sodium ion channels opened to activate the voltage gated sodium ion channels. What does voltage gated means? That means that they get activated by a change in the voltage of the axon. Okay, so normally an axon is a negative inside, okay, and it has more sodium inside. Okay, while it has more potassium outside. Okay, so when the threshold is reached, the voltage gated sodium ion channels will open. Okay, that means that sodium in this part of the axon will keep moving inside. Okay, so if any positive ions keep moving inside, that means the polarity will change. So instead of inside being more negative, now the inside will be positive and outside will be negative. Okay, so this is, if we looked at the curve, the membrane potential will keep increasing and increasing. This flip in polarity is called depolarization, okay, because we changed the polarity. Okay, this increase will keep increasing up to positive 40 when the membrane reaches positive 40. When it reaches positive 40, now all the sodium ion channels will close, okay, and the potassium ion channels will open. Since the potassium is more inside, that means that the gradient will move it to the outside, okay? So now the membrane will dip back down. Okay, will keep decreasing, and it's even decreasing below the resting, okay? So this is called repolarization. We return the polarity again. But here we did not return the original ion gradient, right? Because now we have inside sodium and we have outside potassium. Okay, so we fixed the charge, but we did not fix the ion distribution, which we're going to go to that later. And this dip below the resting, okay, goes up to negative 90. It's called hyperpolarization. We decrease the membrane potential even lower than the original threshold. Okay, so now we have two problems. We have that the membrane potential is lower than the normal, and that it's also the distribution is not right. Okay. This happens because there is more potassium ions than sodium. The membrane is more leaky to potassium, so more potassium leaves than the sodium entering. The resting potential maintained, and how does it return back? So in the membrane, we have something called sodium-potassium ion pumps. Okay, those work with ATP, those to use energy. So in the membrane here, those sodium ion pumps, okay, they pump 3 sodium ions outside. Okay, so those sodium ions go outside, and then 2 potassium ions go back inside. Okay, this fixes the membrane potential back to the resting. And at the same time, to keep the membrane like negative and keep the inside more negative than the outside, there is the leaky potassium ion channels kind of keep working. Okay, that will keep continuously like having potassium moving outside. Okay, so because of this leak, the membrane stays negative from the inside at the resting. What's the function of this part? Like, what's the function of this part over here? Okay, this part is called the refractory period. Okay, this part confuses a lot of people. Okay, so let me like try to clarify it. So the refractory period essentially has two main functions. Firstly, it prevents the backflow of the action potential, and it also sets a maximum frequency for the action potential. Okay, so how does it prevent the backflow? So over here, when we look at this axon here, we said that it starts off by depolarization. So when we depolarize, the inside becomes positive while the outside becomes negative. So now we can say that this section of the action potential is depolarized. Okay, now some of those sodium ions will diffuse sideways, okay, and cause this part. So now this part is also depolarized, okay, and the part before it goes into repolarization. Now the inside is negative and the outside is positive. So what prevents the sodium ions from moving backwards? Why does the action potential keep moving forward? Okay, so it's actually because of two things. So in the first part, before we go below the resting, so in this part, okay, this is called the absolute refractory period. In this period of time, the sodium ion channels had just closed, okay? They stay in an inactivated state, okay? And this time, the sodium ion channels just simply cannot open again. So the sodium cannot go backwards because there's no more channels for it here. So it has no option but to go forward. Okay, and after then, during the period after, starting from the hyperpolarization here, the sodium ion channels are activated again. So now they can open. But the thing is, the membrane is so below resting, it's at -90, that it's going to be so difficult for a threshold, for a stimulus to raise it to the threshold. Because, like, imagine, normally we're at -60, okay, and the threshold is -55. So it's kind of easy for a stimulus to increase it. Like, it only has to increase it 5 millivolts. But if we're at -90, and the threshold is still at -55, that means there is so much more sodium it needs to go upwards. Okay, so it's going to be very difficult for a stimulus to cause the action potential, and so only a very strong stimulus will be felt. So we prevent backwards flow, and we set a maximum frequency, because now we have to wait till the action potential is fully done, and then here we can start doing another action potential. So this sets a maximum frequency and prevents the sensory overload, prevents an increase in action potentials and damaging to your neurons. We set an action potential in the beginning of the axon. How do we move it downwards? How is the transmission? Okay, so we finished the generation. Now we have the transmission. So if we moved like block by block here, like we did here, it's going to take forever to reach the end of the axon. Okay, that's why we have the myelin sheath, as those are called the myelin sheath. The myelin sheath are insulator made of fat, okay, made of Schwann cells actually, and they prevent the sodium ions to move here, so it can't move here. So they only have this empty place, which are called the nodes of Ranvier. Those don't have any Schwann cells, okay? So now the sodium ion channels, let's say the action potential started here, okay, they only have an option to jump up till here. They cannot move here in the middle, so they're gonna jump a big jump till here, and then jump again to here. This is called a saltatory conduction. Okay, this is the jump it's going to jump. And the importance of this is just simply speeding up the transmission, because instead of moving like bit by bit like we can imagine, no, it's going to jump a big jump. So this is called the local circuit. It's going to make it longer. So we have a longer local circuit. Look back here. We saw how an action potential is generated and how a resting potential is returned, and the importance of refractory period and the transmission. Now we have to take the cholinergic synapse. Okay, so now the transmission reached up till here, till the end of the neuron. How did it jump from this part to this part? Here we have to use some chemicals, okay? So let's zoom in here, okay, and take a look. This is one neuron, and then this is another neuron, okay? So the action potential reaches up till here. So we call this the presynaptic membrane and then the postsynaptic membrane. So this part becomes depolarized. Okay, so when the presynaptic membrane becomes depolarized, this causes calcium ion channels to open. So now here, calcium ions will diffuse in. Those are also voltage gated, as we said, because they are controlled by the depolarization. Okay, so when the calcium enters, okay, this causes the vesicles carrying neurotransmitter. Okay, we used to say neurotransmitter in O level, but here they're called acetylcholine. Like the substance inside is called acetylcholine. Okay, so the vesicles containing acetylcholine will move to the presynaptic membrane and fuse with the membrane and then release those acetylcholine in the synapse. Okay, so now they'll diffuse into the synaptic space till they reach the postsynaptic membrane. And now we have obviously receptors on the postsynaptic membrane that are complementary to them, and they will bind on the receptors. When they bind on the receptors, they cause sodium ion channels here to open. So now sodium ions will diffuse in and cause the postsynaptic membrane to depolarize as well. So now we have transmitted the action potential from here to here. After the action potential passes, we cannot keep the acetylcholine here forever, because this means this neuron will stay depolarized forever. So we have to somehow remove this acetylcholine from here. So there's an enzyme called the choline esterase enzyme. This enzyme comes here, fits on the acetylcholine, and removes it. Break it down into acetate and choline. Okay, the choline diffuse back into the presynaptic membrane and go into the mitochondria, okay, to combine with the acetyl coenzyme A and form acetylcholine again, okay? And at the same time, the calcium that was here will be actively pumped outside again. So now this can stop being depolarized and this can stop being depolarized. Function of the synapse, why not just connect it immediately? So we already know the first two from O level. Firstly, it slows down the impulse, okay, which is helpful for your neurons so they don't overload. And it also allows the passage in one direction, because the vesicles are only in the presynaptic neuron and then the receptor are only on the postsynaptic neuron, so it cannot move backwards. Now for the 2 points, which is an interconnection and integration. Okay, so interconnection means that you could have one stimulus that can cause multiple reactions, and vice versa. You can have multiple sensory things that can cause one reaction. So let's kind of simplify this. We have 2 things. We have something called divergence, and there's something called convergence. Okay, so let's say you were walking in the street and you saw a dog. Okay, you saw only one stimulus. Okay, but now multiple parts of your body have to work. So that means that this part will have to synapse with multiple neurons to make your legs work, your heart work, your lungs work, your pupil dilate, multiple things in your body happen. So this is called a divergence, the synapse will with multiple neurons. And the opposite thing can work too. Let's say you were walking into a room and you saw the window open, you saw the lights open, you saw something on the carpet, and you saw something missing for your closet. So now your brain all connects them into means that there was a thief in your room, for example. So all those different information were all connected to the same neuron that made you okay, this means someone is in my room right now. Okay, so this is called convergence, where you converge multiple stimuli into one. Okay, so those are the connections done by your synapse. Something also called integration. Honestly, those points sometimes are written as one, but I just want you guys to understand them just in case. Integration means that sometimes to prevent an overload, if the postsynaptic membrane did not get depolarized enough, the action potential just dies. It does not reach the postsynaptic membrane. Okay, last thing is learning and memory. Honestly, this part is not that learned, we don't know that many details about it, but in general it's known that all your memories are stored as vesicles and synapses. Okay, so when, for example, you're thinking about something right now, you activate a synapse to remember this piece of information. A part that all of you guys hate, although it's not that hard either. So firstly, let's kind of understand the structure, which this I think is a bit difficult to kind of grasp, but let's look at this. This is one muscle fiber, okay? If we zoomed it in, it will give us this one, okay? This was one muscle fiber. So if you look at a muscle, imagine if you're eating a piece of meat, you'll see that it has multiple strips like this, Those are the strips here. Okay, so here we're looking at a bundle of fibers. Okay, so if we looked at it from far away, it's like any like cell. It has like multiple nuclei, obviously, because this is not made of one cell, so it's multinucleated. Okay, it has the outer cell membrane, which is called a sarcolemma. Okay, and the inside cytoplasm is called sarcoplasm. Okay, so it's just a different namings. Okay, and then it has some extra things. So if we looked here, we're gonna see that one muscle fiber has those things here, those yellow things. Those are called the sarcoplasmic reticulum, and next to them they have the T-tubules. Okay, and the sarcoplasmic reticulum have a lot of calcium in them. Okay, and they run very close to the T-tubules. And now with them you're seeing those dots or the striations. Those striations are the myofibrils. Those is where all the contraction happens. Okay, so like each muscle fiber has multiple myofibrils. Okay, so if we take a closer look at one myofibril, we're gonna see this. Okay, it's made up of something thick and something thin. This thick part is called myosin, and this thin part is called actin. Okay, the actin is a thin globular protein, okay, made of many alpha links together, alpha chains, and the chains are kind of twisted together to form a thin filament. Okay, so this is this filament. And the thick part is called the myosin. It's a thick fibrous protein with a globular head. So this is the globular head, the one you can see here. Okay, this globular head is what that has ATPase that we're going to use later. We have a lot of mitochondria because we need a lot of energy. So if we kind of took a crooky drawing of this, because we're obviously not going to draw it perfectly, we're going to have this. Okay, and this is usually what comes in the exam. So to kind of think about it, this part is this part here. So this is the myosin, the thick part. Okay, and then thin part is the actin. So we have to name a couple of things here. The thick part, okay, all this thick part, okay, overlaps with a little bit of the thin part, which is the actin. Okay, this part is called the A band. Okay, this A band has all of the thick myosin and a little bit of the thin myosin. Okay, so this is the A band. Then we have a part in the middle, which is only the myosin part. Okay, so it does not have all of the myosin because there's a little bit that overlaps, but just this part in the middle is called an H band. Okay, and then we have the I band, which is the little part here, which has only the thin part. I know this is confusing, but also just memorize it off the diagram. Okay, it'll kind of just make sense on the diagram. Okay, and then we have this in the middle, it's called the M line, and the one here on the sides with only the actin is called the Z line. So the distance from one Z line to Z line is called the sarcomere. And this is the shortest functional or the smallest functional unit. Like this is the smallest part of the muscle that can contract. This complicated relationship, how does a muscle contract? So it's as easy as this. It just keeps on doing contractions with going back, contractions and going back. That's why when you look at someone contracting their muscle, it kind of keeps shaking because it's not just permanently contracted. It keeps contracting and moving back. So how does this happen? Normally, when there's no action potential, when its muscle is at resting, the actin has the myosin head binding sites, okay? Those binding sites are covered with something called the tropomyosin, okay? So the tropomyosin is a little protein that is twisted around the actin to cover the myosin head binding site on the actin, okay? So now let's say we have our neuron and we have the sarcolemma, okay, which is our muscle. And there is an action potential that moved here and it synapsed and reached the sarcolemma. So now the sarcolemma is depolarized. Okay, so when this is depolarized here, we have in the sarcoplasmic reticulum right here, the depolarization moves towards the T-tubule, so causes the sarcoplasmic reticulum, the calcium ion channels in them to open. Okay, so now there is as we said before, that the sarcoplasmic reticulum has so much calcium ions. So when it opens, the calcium ions move outside. Okay, so when they move outside, they bind here on something called troponin. Okay, the troponin are calcium ion like receptors. So when they bind here, they cause the tropomyosin to kind of move. So when it moves, the binding site on the actin is free now. So now the myosin head can bind. okay? But this myosin head binding site has to be activated. It has to be activated with ATP, okay? So the activated myosin head binding site, myosin head will bind on the binding site, okay? So this is number 1, which is the cross bridge formation. We formed the cross bridge, the actin and myosin bind. Okay, so now this activated ATP with the ATPase in the myosin head will be broken down into ADP and PI, and the PI and ADP and PI will get released. This like shift in energy will cause the myosin head to pivot, will move towards the center. So let's look here. This will pivot like this. Okay, so when this moves like this, it will cause this actin to get pulled inwards. So if we looked at the simplified diagram here, the actin moving inwards, okay, it will shorten this distance right here. Okay, this distance that we called H-band. This distance will get shorter because it will move like this. Okay, same thing with the I-band, because the I-band will also come like this. The only thing that will not change is the A-band, because what moves is only the actin. The myosin kind of stays the same. Okay, so which ones change are only the H and the I. Okay, so the H and the I shorten. Okay, so when they shorten, this is called the power stroke. Okay, now after the ADP gets released, okay, a new ATP will come and bind. So when this ADP will come and bind, it will cause a temporary detachment. Okay, so this is called the cross bridge detachment. Okay, this causes the myosin head to move backwards. Okay, this does not mean that the muscle relaxed. This is a misconception. No, this still happens while the muscle is contracted. This is why the shake keeps happening, because the muscle keeps power stroking and detachment, power stroking and detaching. Okay, and then again, the ADP will get broken down and we will start over the cycle again. So that's why it's called a cross bridge cycle. It's a cycle. Okay, so this will keep happening till the neuron will stop getting a transmission. Once the action potential stops and you no longer need to contract this muscle, okay, the calcium ion channels here will close, okay, and there is something called calcium ion pumps, which those need energy. They pump back the calcium into the sarcoplasmic reticulum. So that's the difference between the calcium ion channels and the pumps. This is something very important. The calcium moves outwards by diffusion, and this happens when there is an action potential. But when there is no longer an action potential, we require ADP to pump back the calcium. This is why when there is no ADP, the muscle cannot relax. Okay, so once the calcium moves back, okay, it will no longer bind on the troponin. This will cause the tropomyosin to come and cover back the binding site. So now the actin can no longer bind to the myosin, and we have our relaxation. The question was like, contrast the structure and function of sensory neurons and motor. So there's a difference between compare, contrast, and similarities. So compare means you say both similarities and differences. Contrast or differences, or just differences, obviously, and similarities, only similarities. So here he wants contrast, structure, and function. Please, guys, please read the question well and make sure to like split up the question. So here you have to talk about the structure and the function. The structure, let's start with the function. The function of the motor neuron is obviously the transmission of the action potential from the CNS to the effector, which is either a muscle or a gland, while the sensory is the transmission of the impulse from the receptor till the CNS. And the CNS, the one in the middle, is the relay. Okay, so just so you understand. And now for the structure, okay, we're just going to look at a motor neuron. So a motor neuron just looks like this. Okay, the cell body is at the beginning of the axon. Okay, and the cell body is usually in the brain or the spinal cord, and it has a long axon and it has dendrites attached on the cell body. Okay, and it has no dendron. While the sensory, the cell body is usually in the middle, okay, and it has a dendron in the beginning and then an axon, and the axon is shorter than the motor, okay, and the dendrites are attached to the dendron, not to the cell body. Okay, and the cell body is not in this CNS. No, it's in something called a dorsal root ganglion. Okay, something not really like directly written, but like it was written in the textbook, so just some extra information. Now here it says that mammals have many types of neurons which vary in axon diameter and myelination. So here we have the myelination and we have the diameter and accordingly the speed. Okay, so something you're supposed to know is the bigger the diameter, the more the speed. Okay, this is kind of like electricity. The more the radius, the more the speed. Okay, so here using the data shown in the table, comment on the relationship between myelination and mean impulse and axon diameter and mean impulse. So when he tells you comment on a relationship, you do not have to describe everything in the table. You simply have to compare those two things together. Okay, so here myelination and mean. Okay, so let's look here. We're gonna see that everybody who is myelinated, okay, has a faster impulse than the unmyelinated motor C. Okay, so our comment is simply going to be that no myelination is slow and myelination is fast. And then we're going to say obviously from the data that motor C, which was unmyelinated, had the least speed of 0.5 to 2, while A, B, and delta, or alpha, beta, and delta, all had faster diameter, okay, faster speed. Then, so now we finished the first point. Second point is axon diameter. So let's look at the axon diameter. So this had the most diameter here, while this had the least. So this had the most speed too. So we're going to say that as the diameter increases, as the speed also increases. Okay, and we're also going to say data from the graph. So it's as simple as that. Do not overcomplicate this answer so you don't end up wasting time and losing marks, okay? So this question was a bit needed some thinking. I was tell us why there is no further action potential during A and B, okay, which is depolarization and repolarization, and why it's difficult for a further action potential to occur at C, which is hyperpolarization. So again, once there is multiple points, we always have to split our answer. So first part is why there is no during A and B. So remember when I explained to you guys that during B, this is called an absolute refractory period because those sodium ion channels are inactivated, okay? They can no longer open till the resting membrane potential. So that's why during B, we cannot open the sodium ion channels, okay, because they're inactivated. And during A, the sodium ion channels are already all open, okay? So this here means that you cannot do another action potential because we're already in one. All the sodium ion channels are already open. We cannot open any more further, okay? So that's the first two. And then during C, okay, we can have an action potential, but it's less likely to occur because of the membrane potential is below the resting, so it's difficult for action potential to get generated, okay? Now for this question about the chemoreceptor, so you have to memorize the chemoreceptor. I'm gonna explain it here so you guys know it. Okay, so firstly, we have this here, Y, that's microvilli, okay, and those here are tight junctions. You can see them labeled. So what are the function of those? They prevent ions to move between the cells. Okay, so now we have chemoreceptor A responds to sodium in salts. Describe how the contact of cell A and it can cause an action potential in B. So this is a type of receptor that detects ions, okay? It detects the sodium when you eat it. Once you eat the sodium directly diffuses in, okay? So unlike our normal receptor, there is no stimulus transduction. No, the stimulus is already what diffuses in, okay? So when the sodium diffuses in, the cell membrane becomes depolarized, and now we have our receptor potential, okay? So if this receptor potential was high enough to exceed the threshold, okay, the voltage-gated sodium ion channels open. Why? Because now we have to talk about the synapse between the receptor and the neuron, okay? So the voltage-gated sodium ion channels here open in the presynaptic knob, okay? And sodium ions diffuse into the presynaptic knob. Now vesicles carrying neurotransmitter, here we cannot say acetylcholine because the neurotransmitter and receptor is different, so we just say neurotransmitter, okay? They fuse with the membrane and exocytosis happen, and then they bind on to the receptors on the postsynaptic membrane, and then the sodium ion channels diffuse in, and now the neuron is depolarized. Okay, so essentially when he asks about the chemoreceptor, you just describe the first part about the sodium and then the part about the synapse. It's very easy. Some students forget to memorize this and they end up losing marks, but it usually comes with 7 marks, and it's a very easy 7 marker. Okay, same here. Okay, this is another question, not the same question. And he also said, explain how the release of calcium ions will lead to an action potential being generated in the sensory neuron B. Okay, so here he kind of skipped over this part, and you just had to describe the synapse part. Okay, so also read the question so you don't just write extra information and waste your time. So this was a bit of a weird section, okay, although this picture was in the textbook, so please guys make sure to look at the textbook diagrams, they are important. Okay, so this is an axon in the middle. Axons look dark in microscopes pictures, and the myelin sheath looks light because it's fat, so it does not stain, so it's usually white like this, and the dark part is the nucleus. Okay, so here, with reference to the voltage-gated ion channels, explain the difference between the myelinated and the non-myelinated. We just talk about the saltatory conduction here. Okay, and this was another question that was asking about a neuromuscular junction allows the transmission of action potential from motor neuron to a striated muscle, causing it to contract. Okay, and this is the action potential in a motor neuron. Okay, and this is an action potential in the striated muscle itself. Okay. Those graph questions have been coming lately, where they get two different types of graphs and then tell you to compare it. Okay, so with reference to the two figures, describe the difference between the action potential in the motor and in the action and in the striated muscle. So first things, we have to look at the resting. This is how you go smart about it. So look at the resting. Here the resting was at -70. Here it was at -90. The resting in the striated is lower. Okay, and now we look at the depolarization. So here the depolarization was from -90 till +40. That's a long depolarization. Well, here it's from -70 to +40. So it's a shorter depolarization. So as a result, it's going to be a shorter repolarization and hyperpolarization. While here, it's longer. Okay, and actually here there is no there's no hyperpolarization here, while here there is a hyperpolarization. That's something also. We calculated the length of all of it and how many millivolts, and then also the time here. Okay, so you calculate from 0 to 6, while here it was from 0 to 3, so it was shorter. Okay, it's quite easy. So here the question was that there are 3 phases in the contraction of a striated muscle. There is the latent phase, there's the contraction phase, and there is the relaxation phase. Okay, here we have a tension graph. Okay, so with reference to figure 9.3, explain what is happening in the striated muscle during the latent phase. So this is the latent phase here. Okay, so there is a stimulation and there is a kind of gap. Okay, there is no contraction yet. So this is the time taken for the myosin and the actin to prep. Okay, this is the time taken for the depolarization, okay, of the T-tubules to happen, and then the calcium channels to open and the calcium to diffuse outside. Okay, and the calcium binding on the troponin and the tropomyosin moving and exposing the binding site for the myosin and actin to bind. Okay, and then the myosin and actin-myosin binding before the power stroke. Okay, so this is the latent phase. The contraction phase is the power stroke phase, when the myosin moved. Okay, then suggest why the relaxation phase shows a gradual decrease in muscle tension. Okay, it's because the cross-bridge cycle break down individually. Each myosin breaks down individually, so this takes time, obviously. Okay, this question was also it seemed hard, but it's actually quite easy. It's showing us 3 states of channels. Okay, so this one is the voltage-gated sodium ion channels, and this is the potassium. Okay, so here he wants to describe what happens during A. So during A, both of them are closed. Okay, so we're going to say state 2. And then during C, okay, that's the time where the sodium ion channels are open and the potassium are not yet. Okay, so it's state 1. Okay, now we have E, which is during repolarization, and this is the time where the sodium is closed and potassium is open, so that's 3. Okay, same was F. Okay, and then G, we're back at resting, so it's 2. Okay, describe and explain the role of myelin sheath. Okay, this is the same thing with the saltatory conduction, that the Schwann cells wrap around the myelin sheath, insulating them. So preventing the ions to move here and preventing depolarization to occur at this place. So now the depolarization only occur at the nodes of Ranvier, making the local circuits longer and the whole subtatory conduction thing, and the speed is faster, up to 100 meter per second. Okay, so that is very important, and the keywords here are important, so do memorize like all the keywords here. Okay. Another question about muscle, it was like that there is a please take care in this question, because this question personally confused me in my exam. Okay, that there is a toxin in this animal. Okay, and the toxin inhibits a protein called the calcium ATPase pump in the sarcoplasmic reticulum. Okay, and it told us here that the pump pumps calcium from the cytoplasm into the sarcoplasmic reticulum when there is no longer a stimulation. Okay, a lot of people, including me, thought that by this pump they mean the calcium ion channels. Okay, so I kept describing how there will be no contraction. Okay, because like the calcium ion channels will not open, so the calcium will not move outside, and there will be no contraction. But it's actually the opposite. It's not the channel, it's the pump. The pump pumps back the calcium from outside to inside. Okay, so this means the calcium will stay outside and it will stay on the troponin and it will keep contracting. There will be no relaxation. Okay, so something very important you have to notice, that there's a difference between the calcium ion channels not opening and the calcium ion channels pump not opening. Okay, so in this case, the calcium channels, the calcium ions stay outside. Okay, they stay bound to the troponin, and the tropomyosin does not move from the binding site, and the actin and myosin will remain contracted. So this will leave the animal in a state of contracted paralysis. Okay, so describe the role of calcium ion channels in the cholinergic receptor. So calcium ions obviously are needed for the vesicles to move outside. So honestly, this is an easy part. Okay, here he wants us to label actin filaments. So this is the actin, it's the light part. Okay, so that's P. And then he wants us to label the myosin. It's the dark part here, which is the R. And as I said, the HAI, the HI contract or get narrower, where the A stays the same. Okay. And here is telling us that the plant Strychnus, whatever this is, has a toxin which causes muscle paralysis. And this toxin acts by binding on the receptor on the sarcolemma at the neuromuscular junction. Okay, so now at the neuromuscular junction, okay, if this toxin is bound, okay, this will compete with the acetylcholine, which was supposed to bind here. So the acetylcholine can no longer bind, so the sodium ion channels will no longer open, and there will no longer be depolarization, so less action potential, and there will be no contraction of the muscle here. Okay. And then suggest why the action of Curie may lead to the death of the animal. Okay, this was a bit confusing because some students kept describing this part again, while he simply just wants the action that caused to the death immediately. So just say any muscle that can't contract and causes the animal to die. So maybe the muscles of the respiration or the muscles of the heart will no longer work, or the animal can no longer escape or can no longer find food because it can no longer move. This question was saying is that a striated muscle will contract when stimulated by a motor neuron. Air temperature can affect the striated muscles. Okay, that they found out that the temperature decreases. When the temperature decreases, so do the contractions. Okay, so he wants us to suggest why. Those are the types of questions that you have to think about. So what does temperature affect? Okay, so temperature usually affects two things we know. It affects enzymes or proteins in general and the channels. So those are the two proteins that we know. Okay, so firstly, any process that required enzyme will decrease. So any enzymes needed for muscles or any respiration will decrease. Okay, and at the same time, the channels will also not work well. So there will be reduction of the movement of ions. Okay, so anything that requires ions will decrease. So like the neuromuscular junction or the sarcomere or the cross-bridge attachment and detachment, all of those will decrease because kinetic energy decreased, so there's less movement, so less work of proteins. Part about plant coordination. Okay, so firstly we have the part about the venous flytrap. This comes like very rarely, but when it comes, it also has a lot of marks, so do know this just in case it comes to you. So for the venous flytrap, those plants, they feed on like bugs and stuff. So we have to know, how does this trap close? So venous flytraps are usually red from the inside to attract insects and green from the outside, okay? And so let's imagine that this is the inside, and this part in the middle are called hinge cells, okay? So they're at the hinge, so they kind of close, okay? So now we will imagine what will happen. So some sort of insect will come here, okay? So on the inside, they have two hair cells, okay? So once an insect touches one of the hair cells, we have a receptor potential. Okay, so the same like what happens to us. If this fly touches the same receptor within 35 seconds or touches the other receptor, okay, an action potential will be generated. Okay, why does this happen? This prevents the same thing like us, the sensory overload. This prevents the plant from excessively closing, because sometimes rainwater falls and the plant could mistake the rainwater for like an insect. So like there's no chance that the rainwater will fall on the same like part twice within 35 seconds, but the insect will. Whatever. So now once an action potential is generated, okay, the action potential spreads on the whole leaf, okay, and reaches till the hinge cells. So when the action potential reaches the hinge cells, this stimulates the production of auxin. The auxin fits and binds to its receptor, stimulating the pumping of protons. This is similar to what happens in the stomata opening and closing, okay? So now the protons will get pumped into the cells, okay, lowering the pH. This dissolves something called calcium pectate. This is the glue. So this is the glue that keeps the hinge cells kind of stuck together. So once this glue is dissolved, now it's easy for once the cell expands to kind of become turgid, okay? So this is the first thing where the glue dissolves. Second thing that happens is that free calcium ions diffuse into the hinge cells. This lowers the water potential gradient, so now water also moves inside, okay? So now we have cells that do not have glue anymore and water that makes them turgid. So this causes this whole, like, we call this a loop, okay? The loop to become turgid and to close like this. So it changes from convex to concave, okay? Because now it's kind of closed like this. So after the venous flytrap, we're going to take how the axon help cell wall, like the cells to expand, okay? How does that happen? So here we have a little cell. This is the cell wall, okay? And this is the inside, the cytoplasm, and this is the membrane. So we have a zoomed in section here. So the axon comes and binds on its receptor on the cell surface membrane. When this axon binds, it causes the ATPase proton pump to pump protons into the cell wall, okay? So now an increase in protons will cause a decrease in pH. Okay, this decrease in pH will cause the activation of expansin proteins, okay, that will loosen the linkage between the cellulose fibrils in the cell wall. This is similar to what happened in the hinge cells of the venous flytrap, okay? So when those cellulose fibrils, like the bonds break, they move past each other, okay? They can move past each other. So now, at the same time, the sodium channels here open. This causes sodium to enter into the cell. So now sodium entered here, so will water through the aquaporins because of the decrease in the water gradient, okay? So now water enters here, the cell is turgid. So because of the cell wall being looser now, those cells, as I said, can move past each other and the cell can expand. Okay, so now the cell expands. Okay, because without this, the water entering will simply make the cell just fatter, not longer, because of the cell wall. But now we loosen the cell wall so the cell can expand. Okay? The part about ghrelin. The problem with ghrelin is it's kind of distributed in multiple parts in the curriculum, and it kind of confuses you sometimes when the question comes. Okay, so ghrelin is in 3 parts. Firstly, it's in the seed germination, in both how like ghrelin is important and the control of the ghrelin. Okay, so importance and control, those are 2 separate questions. And then we have it in stem elongation and then in the rice question, okay? So I'm going to tell you what to write in each one of the questions. So firstly, what is the importance of ghrelin in seed germination? Okay, so when the seed is planted and we add water on the seed, okay, the water activates, is absorbed by the embryo, and the embryo is activated, so it starts the production of ghrelin. Okay, so now ghrelin is produced by the embryo. This ghrelin will diffuse into the alveolar layer, which is this outer layer here, and it will start switching on the genes, okay, for hydrolytic enzyme production. This is the part about the control. Okay, so when the question is how is like ghrelin used in control, or how does ghrelin activate the genes, this is where we're going to say the answer about control that I'll say later. Okay, but if the question was just about importance of ghrelin, so we're going to complete the answer here. So we're going to complete and say that the hydrolytic enzymes will be produced. So the enzymes are like amylase, protease, and maltase, and lipase. So, and then we're going to say each one what will it break down. So the amylase will start breaking down the starch, and then the maltose into glucose, and this will be used in respiration and energy release, and then later in the translocation and building up the cellulose and all this stuff. Protease will be hydrolyzed into amino acid that will be used for growth of the embryo and protein synthesis, and then lipase for growth also and cell membrane production and stuff. Okay, so this is the role of ghrelin. Then how does the ghrelin control? When the ghrelin diffuse into the alveolar layer, how does it activate the genes? So we zoomed in on the cell. This is the cell, and this is the nucleus in the middle. So the ghrelin binds on the complementary receptors on the cell surface membrane outside, okay, and changes the shape and stuff, and it activates enzymes inside the cell. Okay, this enzymes will enter to the nucleus and break down the DELA protein. So who is the DELA protein? The DELA protein is a repressor protein. Okay, the repressor protein keeps binding on the PIF transcription factor, okay, and prevents the transcription factor from binding on the gene and then activating the gene. So as long as the DELA protein is bound to the PIF, the gene will remain unexpressed. Okay, so once those enzymes break down the DELA protein, okay, the DELA protein now, the PIF now is free to go. It's free to move to the gene, okay, and binds on the gene, okay, and then allows the RNA polymerase to move and messenger RNA to get synthesized and transcription of the gene to occur. So now this gene codes for amylase, and there's another one that codes for protease and lipase, so that it can then work here and complete. So as I said, if the question was about the importance of ghrelin, we're going to mention this part. But if it's about the control or how is it switched on, then we talk about this part. Okay, so this is the role in seed germination. So now for stem elongation, there is normal plants and there's something called dwarf plants. Okay, those dwarf plants, they do not have the active form of the ghrelin. So the active form of a ghrelin is a dominant trait. So it has, you have to know this, has a L capital E. So that's the homozygous dominant. Okay, people will have the recessive L small e. Okay, those people are dwarf plants. Okay, or not people. Those plants are dwarf plants. Okay, they do not have active ghrelin. So how does this affect the plant? So this active ghrelin was supposed to activate enzymes that cause cell elongation. So kind of the same thing that happens here, okay, with the whole DELA protein and PIF and stuff. This active ghrelin was supposed to go and activate the genes that synthesize the enzymes responsible for cell division and cell elongation. So if the plant does not have this active ghrelin, those enzymes will not be switched on, so the plant remains as a dwarf plant. Okay, this can be helpful sometimes. So we sometimes cause this mutation. Why? Because sometimes a plant uses up its energy and its food to grow. Okay, and we do not get use from this. We want the plant to have more food value, so we want it to be shorter. So it uses up all the energy into making more grains and making the grains fatter. Okay, so this is sometimes useful. That's why we sometimes make dwarf variants ourselves. About ghrelin is in the respiration topic. It's about the rice plants and how it is useful here. So in rice plants, they are grown obviously in water. Okay, so this causes an increase in aerobic respiration and an increase in ethene. Okay, so this increase in ethene is actually helpful for the plant because when the ethene increases, this activates the ghrelin hormone also. Okay, so this activated ghrelin hormone, as we said before, increases cell division and cell elongation. Okay, so now this part of the plant is called an internodal length. Okay, so when this gibberellin causes more cell elongation and cell division, the internodal length increases. The plant becomes taller. So this is helpful for rice plants because, as we said, they're in water. So if they remain low, they cannot reach like sunlight for photosynthesis, and they will not be able to produce because like no pollinators will reach them. But if they increased in length a bit, like they will reach more sun and they can be more pollinated. So this is an advantage and one of the actually adaptations of the rice plants.